So in today's section, we will discuss about blood group, and we also call this blood group as a ABO system.
Hmm.
Why ABO? Because all four different types of blood group are represented in this notation: A blood group, B blood group, as well as AB and O blood group. There was a scientist called uh, Carl Landsteiner. He has discovered this system. So this blood group system discovered by Carl Landsteiner in 1900. So I guess 121 years back. Okay, and this system, this ABO system or the blood group system, was actually based on presence or absence of an antigen which is present on RBC. Right, so you can say that it is this group. Group means blood type, are based on presence or absence of antigen present on RBC. And if there is an antigen, that means there must be an antibody, right? So
Yeah.
you can say that and presence and absence of antibodies antibodies in plasma. So these are the two ways in which you can say that blood group is based on these two factors. Either the antigen, you can talk in terms of antigen present on RBC, or you can talk in terms of antibodies present in the blood plasma. So ABO blood group is actually, if you'll go inside this antigen and you are trying to find out, then you will find that it is based on presence and absence of two surface antigen. So if I have to talk about the antigen here, so let's talk in terms of antigen. So there is two surface antigens are present on the RBC. What are the two? We call them antigen A and antigen B. So these are the two broad categories of antigen present on RBC. Fine? Got it? ABO blood group is controlled by three genes or three alleles, you can say here. So, okay, so I have to discuss something about dominant and the recessive character. You have any idea about it? What is dominant character? What is recessive character? What
Yes, sir.
is
I know.
homozygous, homozygous,
Yes.
heterozygous? Okay, good.
Yes, sir. I know.
Codominance,
Yes, sir. I know that as well.
independent assortment.
Yes, sir. I know. I learned all of these terms in grade 10.
Okay, then that's good. Then you will able to understand this. So ABO blood group are controlled by three allele of a gene. So you can say here that blood group are controlled by 3 allele of a gene, and this phenomena is called multiple allelism. Now, if I'll talk about the Mendel's discovery, Mendel has told one thing, that one gene, one character. He has given this hypothesis. This means that one gene always controls one character. Later on, after further research in the genetics, there was a scientist called T.H. Morgan he is also called father of modern genetics, where he has said that there are n number of genes responsible for carrying one character, like skin color, right? Then blood group inheritance. These things are controlled by more than one gene. So if any character is being controlled by more than one gene, that comes under multiple allelism. Fine. So when we say 3 allele, the meaning is there are 3 genes we generally represent as IA, like this, IB, or IO, or you can write small I. In any way, you can represent these uh, O blood group. Now, these two genes are dominant gene, and this is your recessive gene. So you can say IA and IB are dominant, and small I is a recessive character. So now, if dominant gene are present, that means the dominant allele are responsible for formation of surface antigen, right? So you can say here that dominant allele are responsible for formation of surface antigen. And we know that there is two types of surface antigen, antigen A, antigen B, right, on RBC. Okay, so let's take a condition. If antigen capital A is there on RBC, then plasma will have antibody B. So you can say plasma will have antibody B, or you can write anti-B. Now, if there is antigen B is there on RBC, then plasma will have antibody A, or you can say anti-A. So an individual can have any two of these alleles, right? So in that way, what are the possible combinations? So let's see. Let me make a table to understand this more better. So I'm here writing blood group. Then we'll write here alleles. Then we'll talk about antigen. We'll say antibodies. Then We'll talk about blood transfusion. So you can say can be given to person with blood group, and the last column you can say can be taken from the person with blood group. Fine. So let's begin with the A blood group first. So if suppose there is A blood group, then the possible alleles which carry this particular character can be represented as capital IA, or you can say capital I and small I, because capital I is the dominant character. If this gene is present, the character will be always A blood group. Small I is the recessive character, so this is not going to be expressed in the generation, right?
Yeah.
Okay. Now, antigen, what type of antigen present on RBC? That is antigen A, right? Now, antibodies on plasma will be small b, right? Now, we'll discuss about the rest two, these two later on. If we will talk about B blood group, so if a person have IB and IB, when both the genes are dominant, then person have B blood group or capital IB and small I. Means father is giving capital IB, mother is giving small I, or anyway, you will get B blood group in the generation. Antibodies will be A. In the same way, AB. AB is a case of codominance. Codominance means when both the genes are expressed in the generation, dominant as well as recessive character. And the genes which controls their inheritance is capital I and capital B. Capital IA, capital IB. There is no other way, right? So both the genes are present and both are equally expressed in the generation. So you will find that the child is AB blood group. If you will talk about O blood group, to get O, you need both recessive character in the generation and they have no antigen. Now, do AB have antibodies? No. But yes, O blood group have anti-A and B both in the plasma, right? So this antigen is present on RBC and this antibody is present in plasma. Now, if you talk about the blood transfusion, so this is your blood transfusion, right? So A blood group can give to A and AB, and who can give blood to A? A can give and O can give. B blood group and AB blood group cannot give blood to the A person due to the difference in the antibodies. Now let's think. If suppose AB blood group is given to A blood group, right? So on A blood group there is antibody B, so they will start destroying uh, antigen present on AB blood group, because this is antibody always attack antigen. Got it? Now if suppose you are giving B blood group to A, so B having antigen capital B and A having antibodies B, so this will interact and they will kill this antigen, right? So that's why on the basis of antigen and antibody, the blood transfusion is So here you can write for B, B can give blood to B blood group and AB. And who can give blood to B? B can give and O can give. What about AB? AB can give only to AB, but can take blood from AB, A, B and O. In case of O blood group, O can give to person, can be given to a person with blood group O and AB. And who can take blood from this group? So you can write A also, B also, right? So all can take. But when it comes to receive the blood, then O can only take blood from O blood group. Is this table is convenient? Can you understand?
Yes, sir, I can understand.
Okay, so let me put a line here to separate. So this is the concept of antigen antibodies present in the blood. So while blood transfusion, these things are checked. So we can say here that this particular blood group AB is acting as a universal recipient, and O blood group is universal donor because O can give blood to any blood group. So you can say that during blood transfusion, blood group matching is essential to avoid antigen antibody interaction, right? If you want that blood should not get clot within the body, should not get uh, coagulated, you must check antigen antibody compatibility. It should not match. If it is matching, then it will cause coagulation. I have not discussed about plus and minus. We say A plus, B plus, or AB plus, or O plus, right? So what is the concept of plus that we will discuss now after this, right?
Sir, is it possible to change your blood group at any point in life?
No, dear. How? Think. Okay, suppose you want to change. Now, so first we have to find out the DNA which is responsible to make these proteins, right?
Hmm.
So, okay, so suppose you have done alteration in one particular cell, right? And you have put it in the body. Then that
Hmm.
cell will produce a different blood group, right?
Yeah.
then what your body will see? Body will take that cell as a non-self cell. Non-self means your immune system recognizes your body own cells, and
Hmm.
body owns cellular activity also. If
Hmm.
anyone is doing weird behavior, right, then there are so many processes like apoptosis, necrosis, programmed cell
Hmm.
death,
Hmm.
or cell suiciding, that's called apoptosis, where an
Hmm.
instruction is being released inside the cell by the same cell to destroy the cell.
Yeah.
Okay, at 1% chance the cell is successfully able to integrate in the body. Okay,
Hmm.
cell starts producing protein. As the protein will release in the blood, there are antibodies are there. They will destroy that particular protein.
Yeah.
So it's
Hmm.
impossible.
Hmm.
So if you want to make sure that blood, see again the problem is, if you want to change, you have to change your mother and I mean your parents' blood group first, right?
Hmm.
Then only
Yeah.
then only these things are possible, because the blood
Hmm.
group genes are coming from your parents. If your parents have such gene, then that gene will come in you.
Hmm.
So there is no chance of, now if suppose the question is, can the next generation will have O blood group, or A blood group, or AB or B blood group, that is totally based on the genes of the parent.
Yeah.
So hypothetically and theoretically, it is impossible to change the blood group at any point of time, right? Not possible.
Hmm.
Okay, so I was talking about the blood transfusion, so we'll write few points here. First point is during blood transfusion, blood group matching is essential even while organ donation. These things are checked. So let's talk about one example. So example one is, if a person with A blood group, A blood group means there is antigen A on RBC and antibody B in plasma, right? That's the meaning. Is given the transfusion of blood of type B. Let's take this example. B blood group is giving blood to A blood group. And in type B blood group, we know that there is antigen capital B and antibody A in plasma. Right? So what will happen? Antigen antibody interaction will take place here. So you can say that the antibody B will react with antigen capital B. So small b is antibody, capital B is the antigen. So they will react, and in the same way and antibody A will react with antigen capital A. This results in destruction of RBC, right? When RBC is destructed, who will carry the oxygen? No one. Person will die immediately, right? So you can say that this results in destruction of RBC reaction, RBC resulting in either anemia and jaundice. So these are the two cases. Such reactions like antigen-antibody compatibility reaction, such reactions between different blood group is known as ABO incompatibility complex. So you can write one more point saying that such reaction between different blood group is known as ABO incompatibility. If you want, you can add such reaction between antigen and antibody. Antigen and antibody. Antibodies of different blood group is known as ABO incompatibility. So ABO incompatibility results in milder reaction like mild anemia and the jaundice. So these are the causes. So you can say here leads to jaundice and anemia. So in the same way, if you are giving blood to B blood group, A or any other organism, you can see the antibody and antigen compatibility. If it is not matching, then you can give it. If it is matching, then it may cause interaction between antigen and antibody and they will destroy each other. And always remember, antigen are present on RBC. So if antibodies are going to destroy antigen, that means you are destroying RBC cell. And we know that RBC is important cell required for oxygen transportation. Sometimes it is very dangerous. Fine. So let's talk about Rh factor. Stop here. You have any idea about this? What is Rh factor?
Yes, sir. I know what it is.
What is the name of this Rh mean?
I don't know if I'm pronouncing it correct, but it's apparently called rhesus factor.
Yeah, rhesus
And
factor. Mhm.
yeah, and it's basically determined by D antigen.
D antigen. Okay. So fine. So Rh factor is a protein which is on present in a monkey, and that monkey called rhesus monkey, and these monkeys have this protein, Rh protein, or also called Rh factor. So based on this factor, whether it's present or absent, we categorize the entire human blood system as a positive and negative. Right? So you can say here first line that another Rh antigen, which is actually detected in monkey. So you can write here detected in rhesus monkey, is also found on surface of RBC. So apart from antigen A and B, you have another antigen present on RBC, which is called Rh antigen. So on this particular basis, Rh factor, if this factor is present on RBC, we call it plus, positive, Rh positive, Rh negative. When protein is absent, we call it negative, and when protein is present, we call it positive. Fine. So if you will take a human sample, sample means like 1,000 people, and you want to check whether this antigen is present in the blood plasma or not. I mean, on not on blood plasma, on RBC, you will come to know that 80 to 85% of the human population have this particular protein present on RBC, and around 15 to 20% of people, they don't have this antigen. And again, the pattern of blood group system is as it is. So it's A plus means antigen A and Rh both are present. In the same way, B plus, AB plus, and O plus. When we say A minus, the meaning is antigen A is there, but Rh is absent. That's why minus. B minus, AB minus, O minus. Now the question is who will give blood to whom? So positive will give to the positive one, but it can't give to the negative one. Any reason why?
Because the Rh positive protein is a dominant trait, but Rh negative is recessive,
No,
right?
the answer is no. The answer is Rh factor, Rh plus means this protein is there, right? When this blood is given to the negative person, then this Rh protein will act as an antigen, and your negative blood group person's body will start making antibody to destroy this particular antigen present in the blood. So again,
Yeah.
it will result in the uh, ABO incompatibility, which will result in some disease. Now the best part is negative one can give blood to the positive one, right? A negative is suppose given to A positive, so no problem. A positive have to work hard to produce more Rh factors, right? So slowly as the blood cells number will increase, you will find that they are being replaced by the Rh antigens. So there is no problem. Body will make Rh protein because DNA is there, right? So negative one can give blood to positive one, but positive one cannot give blood to the negative ones. Now, if you have to tell me which is universal donor here,
O negative.
because O can give to the positive also and the negative also. And which is the
Hmm.
rarest blood group here?
That would also, okay. It's either AB negative or O negative.
Well, AB negative can give blood to A, B, and uh, AB plus, B also, A also. Sorry, not
No,
A
I
and B.
mean it can give, but no, but when you said rarest blood group,
See,
usually
when we say rarest blood group, the meaning is no one can give blood to it. Only the similar blood group person can give blood to them,
okay, then O negative.
right? So O negative will give blood to everyone, but when it comes to receiving the blood, then it requires only O negative person. Fine. A negative, B negative, and AB negative are not a rarest blood group because negative people can give blood to the negative. So there is a chance of blood transfusion here within the Rh factors, right? Hope
Hmm.
this makes sense to you.
Yes, sir. Makes sense.
Right? So we'll write some point here. So you can say that 80% of human population has the antigen. In the bracket you can write RH. So they are called Rh positive, and 20% do not have this antigen, so they are called negative, Rh negative. Again, here also, while the blood transfusion process, Rh factor is considered here. So you can say that blood matching is essential to check Rh compatibility. Next point, you can say that antibody against Rh antigen is generally not present. That's why it becomes very easy to give negative blood to the positive one. So you can say here that antibody against Rh antigen is normally present. These antibodies are synthesized only on exposure to the antigen. So if antigen is present, then only body will produce antibodies. So when you are doing blood transfusion, you must check. Now, if the question is, might be you are thinking, if suppose by mistake negative blood is given to the positive one, or the sorry, positive blood is given to the negative one, is there any chance we can prevent the incompatibility things. So answer is yes, you can, by giving antibodies dose at the same time, so that all the Rh factors will be destroyed. But again, it is life threatening process. No one do this, right?
Mm.
Last point, a special case is when the pregnant female have Rh negative blood, and the fetus in her womb is Rh positive. Is this possible? So yes, it is possible. Mother is negative, but the child which is growing in her inside her body is Rh positive. So is there any problem? So answer is yes. So from the female body, the antibody will develop and it will go into the fetus to destroy the fetus. Now, how to handle this? So many years back, there was no such technique to stop the movement of antibodies from the mother's body to the fetal development sac. Now there are some medication by which we can stop the entry of antibodies. Right? So let's discuss what happens then. So Rh antigen on fetal RBC does not come in contact with the maternal blood. Now, I hope you know that there is no blood connection between mother and the fetus. The mother's blood, suppose this is mother and this is fetus, right? So suppose this is the mother's blood and this is, let me draw as a blood vessels, yeah, and this is fetal blood vessels. So both the blood vessels are near to each other, and the substance which is present in female blood, like oxygen, glucose, amino acid, this is being crossed by the process of diffusing, because fetal blood vessels have less oxygen, less glucose, less amino acid. So due to the gradient, the substance will get crossed, right? And in the same way, the fetal blood vessels have more waste substances, yeah, like urea, uric acid. These are transported into the mother's blood. So you can say here that Rh antigen on fetal RBC does not come in contact of maternal blood. So you can see in the diagram that they are, both the blood vessels are present on close proximity, but there is no direct connection. Right? Maternal blood and fetal blood are separated by placenta. So placenta is a junction which provide that close proximity situation. So placenta is a barrier which prevents the direct connection. But during parturition, mixing of blood takes place. Right? So you can see here that but Mixing of blood takes place and mother's blood or mother's body receives Rh antigen from fetal blood. Then what will happen? Mother's body now prepares antibodies. So next point you can say mother's body now prepare antibody against Rh antigen. And we know that this Rh antigen is present in the fetus. So as in the first delivery, mixing takes place. Mixing of blood takes place for a very short period of time. So when we say short period of time, that means only during paturations, right? So you can say that the reaction is not serious. Short period of time, the reaction is not that serious. So the first child have no problem. The problem is in the subsequent child. So in the subsequent child, pregnancies, Rh antigen from mother blood go to the fetus and they destroy it because
Hmm.
because antibody is now being prepared in the body, in the female body, right? So last point you can say that in subsequent pregnancy, Rh factor, or you can write Rh antibody from mother's blood go to the fetus, destroying the fetal RBC, right? This causes severe anemia. Maybe you have heard when a child is born, child is anemic or sometimes jaundice. So due to this reason and can be fatal. This condition is called erythroblastosis. That's the term given. This condition is called erythroblastosis fetalis. So this can be avoided. There is a way also. So you can say the last point saying that this can be avoided by administering or administrating anti-Rh protein, anti-Rh antibody to the mother immediately after first delivery. So this anti-Rh antibody will prevent the further formation of antibody in the mother. And when a mother is going to get pregnant second time, then these antibodies are not there. So there is no problem. Fine. So this is all about Rh factor and Rh compatibility. And one special case we have seen, pregnancy where the mother is negative but the fetal is positive. How uh, the pregnancy is being conducted in the second case. First case, there is no problem, but yes, in the second pregnancy, there will be a problem. And again, there is a remedy also by giving Rh antibodies to the mother. So that's it for today's section.
Hmm.
Hope you have written all the points.
Yes, sir. I've noted it down.
Fine then. Bye. See you.
All right then. Bye. Good night, sir.
Right.